That was a really hard act to follow. On the other hand, you'll thank me for not trying to imitate. It's a real pleasure to introduce Shaul, not because he needs any introduction here, he certainly doesn't, but because it allows me to express some of my admiration and affection for him and his work. We started in backgrounds that could not have been more different. My first degree was in French literature and Shaul in physics. So nevertheless, I did get a degree in psychology after that, and we managed to come close enough so that we could collaborate on a joint grant for many years. I'm somewhat ashamed to say that we didn't publish any joint papers, but we did have an important influence on each other, shown in the degree to which our theories tended to converge, mainly actually because mine moved towards his very creative idea that consciousness accesses the highest levels of representation first and then moves back down the hierarchy to lower levels as needed to perform the current tasks. Can you hear me at the back? I thought I'd say a few words tonight about the reverse hierarchy theory that Shaul and Merav developed between them, because that's the topic closest to my own research. It's certainly familiar to most of you since this is the birthplace of the theory. And just to remind you, though, the idea, in their words, is that processing along the feed-forward hierarchy of areas leading to increasingly complex representations is automatic and implicit, while conscious perception begins at the hierarchy's top, gradually returning downward as needed. Perceptual learning follows a similar sequence, starting at the high levels with easy conditions, short exposure durations, considerable transfer across stimuli, and with practice, if necessary, it moves down to lower levels with more specificity, less transfer, and so on. It's a very satisfying theory because it's so simple, and yet it accounts for many phenomena that caused problems for the view that was standard at the time. The traditional idea, perhaps going back to Hubel and Wiesel, was that vision goes through a series of levels of representation, starting with simple elements like lines and angles and edges, then to combinations of these, abstracting them from particular contrasts, sizes, backgrounds, and so on, and finally to more complex conjunctions, which represent the familiar objects that we recognize every day. This view is still accepted. There's lots of evidence for it, at least as a simplified approximation to the truth, as Bob reminded us this morning. Certainly the sequence corresponds closely to the order in which brain areas respond to visual stimuli. But the accompanying idea that the same order determines when these levels can be consciously accessed is what Shaul and Merav challenged. There were many problems with the claim that the levels of representation become accessible in the same order as they're computed. For example, the speed of target detection in search is often slower for simpler elements like lines and angles, which presumably would be detected first, than for meaningful units like faces, birds, buses, and so on. The fastest access is often to the features of three-dimensional scenes rather than to the retinally defined features. Repetition blindness is defined at the level of categories, not at the level of literal visual repetition. Yet it took many years, more than 30, I think, for anyone to challenge the assumption that the order of conscious access must match the order of brain processing. Perhaps it needed the widespread acceptance of implicit processing, shown in both memory and priming studies, to break our assumption that whatever the brain computes and makes explicit at any stage will be consciously available as soon as it's neurally represented. So that's why we should be able to get the level 1 things very early. I was guilty of a similar assumption when I was working on selective listening in the 60s and 70s, defending early selection against late selection accounts of attention. I didn't allow for the possibility that the brain might know things that its owner did not. In other words, for implicit processing. I still believe that some selection occurs early, but I think one needs more evidence for it than just people's inability to report the content of unattended messages. I find the general claims of reverse hierarchy theory very convincing, But in order to fill my allotted 20 minutes, I'll add a few comments that elaborate further on some points that they raise. One concerns the binding of features, and the other is about consciousness. Shaul and Mirav explain the fact that search is slow for conjunction targets, for example, a red O among green O's and red T's, by the fact that it needs accurate localization. So in order to prevent illusory conjunctions, we need to check that the target features, the red and the O, are in the same location, belong to the same object. And so we need the small receptive fields that are found in early visual areas. We got some further evidence that conjoining features depends on re-entry, consistent with the reverse hierarchy. DiLollo, Enns, and Rensink developed a type of masking that they called object substitution masking. This consists of, what they did was place 4 dots around the target stimulus, not actually on it, but just around it, and left them visible for a couple of 100 milliseconds after the target disappeared. Excuse me. Unlike regular masking, this disrupts conscious awareness of the masked object without necessarily destroying any other effects that it might have. Their idea was that when the dots remain visible after the stimulus has disappeared, they replace the stimulus at the critical time that re-entry would arrive at the early level. My postdoc, Seth Bouvier, and I showed that this kind of masking leaves feature reports intact. We didn't get any damage from the mask, but it badly disrupts reports of their conjunctions. Brait, Wouter, and Humphreys also used transcranial magnetic stimulation to confirm that conjunctions were selectively vulnerable to disruption a short interval later than features. These findings are very consistent, I think, with the reverse hierarchy theory. It must take some time to go back down the hierarchy to the early levels of vision to find the relevant features to bind together. One question that puzzled me is how we put these findings together with the fact that the parietal lobes are clearly active, specifically in conjunction search. Do the parietal lobes actually control the tracking back down the hierarchy, or are they needed to integrate the separate features once they have been retrieved from the low levels where accurate localization is possible? I think I'd favor the second possibility, but we do need further evidence on that. Another piece of evidence on which the reverse hierarchy relies is the idea that the high levels are accessed not only early, but also in parallel, so that high-level targets in a search task might pop out of a display without any effect of how many non-targets are present. For example, Shaul and his colleagues found parallel pop-out for faces among cars and houses. It certainly fits well with the fact that the features that do pop out are often high-level features, defined as those of a three-dimensional scene on which illusions have already acted. So the Muller-Lyer illusion, that's up in the middle there, changes the apparent length of the center line, and it's the perceived length, not the true length, that determines whether the line will pop out or not. So there's one long line that probably does pop out, which is actually the same length as the other lines in the figure, and there's a short one middle top, which is the target and which doesn't pop out because the illusion makes it look longer than it actually is. One problem with this idea is that the high-level features often have low-level correlates, which might be the actual targets of search. Faces have typical color, curvature, and other properties that could be detected by early neural units rather than the high-level configural patterns that would emerge only at high levels of the hierarchy. This is a really hard problem to control for. One valiant attempt was made by Treisman and Koenig. Their solution was to use specific conjunctions of the features of schematic faces, nose length, eye separation, eye height, and mouth height, as the high-level targets in a search task. They trained people on these, so they got extensive training, 16 sessions, to recognize these composite faces, but they found that the search slopes, when they were looking for one of them as a target among the others as distractors, never became flat, showing no evidence of pop-out for the learned faces or high-level units in this task. And this was supposed to cast some doubt on reverse hierarchy theory. But I wonder if the critical factor here was the fact that the targets were conjunctions and so required feature binding, even though the categories were well learned. And actually, I'm not sure that the reverse hierarchy theory predicts that high-level search must be parallel as well as early. We can ask the authorities. It seems possible that even rapid conscious access must sometimes be spatially serial, even though it starts early after stimulus onset. When the high-level targets require binding, at least according to my account, they should still depend on localization and therefore require serial search. So according to my theory, high-level pop-out, like low-level pop-out, should only be found when the high-level target has a unique feature not also present in the distractors. And that was certainly not the case with K and K's schematic faces. The claim is that pop-out depends on having a unique representation of the target in the display at whatever level. If there are emergent properties that are specific to particular units, pop-out could be possible, since these emergent features need not be accurately localized. It's enough just to say that there is one there. However, K and K's faces were deliberately designed with no emergent features. The other claim that I'll spend a few moments discussing concerns the role Shaul and Mirav envision for consciousness. And here, this is mainly my ignorance that I'm going to show. They talk about consciousness accessing a particular level of representation. This means that we become aware of what is represented at that level. But in what sense? The idea of consciousness sliding up and down the perceptual hierarchy as the task requires is unclear to me. I believe Shaul and Mirav do think of consciousness as moving, at least according to their papers. They suggest that the site of consciousness, if there is such a place, should be thought of as moving around from time to time, not as a fixed area of the brain. This seems to imply that when we're at a particular level, we simply experience the information that's present there. For example, we see a set of oriented edges without meaning when consciousness is at level V1. Do we really lose access to other levels when consciousness moves to a different one? For example, when we're doing a difficult feature search for an orientation among distractors with other very similar orientations, do we lose awareness of the higher level objects that instantiate those orientations? For example, the variously oriented cars in their figure. They have various cars, different tilts. One alternative to the idea of consciousness moving up and down the hierarchy might be that it stays at the high level, where we see organized, meaningful categories of objects, but that we can make available more precise information from selected lower level representations to the high level that occupies awareness. Mirav and Shaul do suggest that the weights of evidence from different levels can change, but I think they tie this to the presence of consciousness at that level. I think I prefer the view that the level of consciousness remains the same, that it depicts our best guess about the nature of the world around us, given the information available, and our current task or interests. What changes is the information that reaches that level. So we still see a variety of oriented cars in their picture, not an abstracted set of lines and edge orientations. Well, I won't bother with the slide because it'll take me a while to find it. But as time passes, and if the task requires it, we become more aware of the precise orientations of the cars. What moves down the hierarchy on this view is the relative weighting of orientation information from early levels that go into the high-level picture. Another possibility might be that the low-level orientations become more finely tuned, but this seems less likely to me. I don't know if there is any evidence that the tuning of V1 units becomes sharper when an animal is conscious than when it's unconscious. I don't know of any evidence on that, but I could be wrong. Perhaps one piece of evidence against the option that consciousness moves back down the hierarchy is the difficulty we have in overcoming certain visual illusions. For example, the illusory odd one out in line length in that Muller-Lyer picture, and the indented square in the bottom picture that can be seen as occluded in depth are much harder to see than the true line length or the unexplained indentation. Does this reflect constraints on what levels we can consciously access, or is it simply that the high levels are difficult to ignore and tend to dominate our awareness? Note that similarly, it's quite hard to move even higher up the hierarchy. There seems to be a level where consciousness resides, at least to my sense. We can't visualize an animal, just a particular animal, or perhaps a rapid kaleidoscopic sequence of different animals. Danny Kahneman and I suggested that the level of consciousness in vision corresponds to the temporary object files that we hypothesized are formed when we see particular currently present objects. Consciousness can certainly manipulate how much specific detail is available in these object files, but I think it's harder to change the level in the hierarchy at which they are consciously represented. I think we see meaningful objects. I'm not sure whether that was what you meant by consciousness moving down the hierarchy, but I had to say something in my 20 minutes. These questions about consciousness in no way undermine the general claims, I think, of the reverse hierarchy theory. As I said, I think it makes a major conceptual contribution to our understanding of perception and perceptual learning. On a more personal note, let me return to Shaul, whom we celebrate tonight. What makes him so special? One is the breadth of his interests. He started with visual responses in barnacles, as we heard this morning, no less. Who would have thought that he'd become a highly regarded cognitive psychologist? His CV spans work on the basic physiology of the motor system, on models of complex cell receptive fields, on multisensory integration in the cat superior colliculus, on classification in monkeys, and finally on human consciousness. Today we heard a sample of studies in many of those regions of interest. It's a wonderful range of topics, in each of which Shaul did beautiful, organized, and incisive studies. He has a gift for seeing the whole picture, creating an integration of all the levels of analysis. I'm a very messy investigator and writer myself, so I really appreciate the clarity and the structure of his presentations. When we were collaborating, he'd come with some beautiful graphs to show me what he'd discovered since the last meeting. Even his beautiful figures and the amount of data he collects in each area really make me envious. Finally, the imagination and creativity of his papers and the simplicity with which he communicates them stand out for me. I was very lucky to have those years of collaboration with him, and I'd like to join you in celebrating him today. Thanks.
Shaul needs no introduction.
Thank you.
But
Okay. Oh, no, I wanted that right.
having known Shaul all these years, I can say a few things. The first is he's a wonderful scientist. The second is he's a great friend. The third is that he's a Talmudic scholar. And the fourth is he is a very wise man. And he's going to tell us tonight about advice to young scientists. And I can think of no one better to do that.
Thank you, Mickey. Thank you, Mickey. Thank you, Anne. I think you noticed from Anne's introduction why I enjoyed so much meeting with her. It was always very challenging and always made me think more and more, even about the things that that I and my colleagues developed. Before I put away this slide, which Anne chose, I just want to point out something that Meirav and I quoted already in this paper from Kierkegaard. Life can only be understood backward, but it must be lived forward. So if any of you think that I'm going to look back, then no, we're living forward, not back. Let me get rid of all this junk and see if we can get to the PowerPoint, which is where we are. Not supposed to do that, but okay. No, wrong one. 1 second, we'll get there. It's supposed to be open. It is advice to young scientists. Here we go. Okay. Okay. Let me also say, just as an opening, wow, this has been quite a day and quite a week. I think the organizers felt that, Meirav felt that since this is celebrating 40 years, we have to have at least 40 talks. And so it was. Actually, the number 40 is a very interesting one. My mother, who passed away just a little bit more than a year ago, was a bit of a Bible scholar herself. And she used to say that the number 40 in the Bible is not a number which is a precise number, but it just means a lot. So in Noah, the rain came for 40 days and 40 nights, and Moses went up to the heaven to get the Torah. It took him 40 days and 40 nights, and the Jews wandered in the wilderness for 40 years, etc. And ultimately, I think it's after Dvorah wins her battle that Vatishkot haaretz 40 shana, that there's peace for 40 years, and that's a long time to have peace, as I think we know here too. So 40 is a long time, and so we can, at this point, look back also to try and understand, and at the same time, think about what we want to do ahead. And the title, Advice to Young Scientists, is not new for me. There are a number of books or essays on this topic. For example, well, let's start with Ramoni Kahal, who says the following kinds of things. I believe that all outstanding work in art as well as in science, and thank you, Karen and Ellie, for bringing music to this event. I know it was a special difficulty for Karen to come and to sing with, etc. She was wonderful. All outstanding work in art as well as in science results from the immense zeal applied to a great idea. Beginner's traps to the young scientists. Undue admiration of authority. Don't. The most important problems are already solved. They're not. Preoccupation with applied science. Yeah, you can do applied science, but perceived lack of one's own ability. To which he adds, defect for defect, arrogance is preferable to diffidence. Boldness measures its strength and conquers or is conquered, while undue modesty flees from the battle, condemned to shameful inactivity. And finally, science is often built on the ruins of theories once thought to be indestructible. I think we've participated in that, I think, on both ends. Major sources of knowledge include observation, experiment, and reasoning by induction and deduction. Now and then, philosophers invade the field of biological sciences with these beguiling generalizations which tend to be unproductive, purely verbal solutions lacking in substance. And if that's not enough, there's no doubt that the human mind is fundamentally, mentally incapable of solving those formidable problems: the origin of life, nature of matter, origin of movement, and the appearance of consciousness. Well, let's move on to Peter Medawar, who's more recent. This is advice to ELSC. Inbreeding is often the way in which a great school of research is built up. Any scientist of any age who wants to make important discoveries must study important problems. Remember. Then he says, twice in my life I have spent 2 weary and scientifically profitless years seeking evidence to corroborate dearly loved hypotheses that later proved to be groundless. My advice to young scientists is to have more than one string in their bow and be willing to take no for an answer if the evidence points that way. I like this image not of 2 arrows in the bow, but 2 strings in the bow, 2 ways of approaching things. Well, enough. Let's go on a word by Mark Twain, just for fun. By the way, Benjamin Franklin's advice is to a young man on the choice of a mistress. He's talking, of course, premaritally. But even so, well, look it up. It's interesting. Mark Twain says the following: always obey your parents when they are present. Be respectful to your superiors if you have any. There are many sorts of books, but good ones are the sort for the young to read. I would say that about journal articles as well. Read the good ones. Okay, so now a little bit of my own advice to you. First, choose to be a scientist if and only if you enjoy doing science. I feel extremely lucky to be doing what I love doing, and even earning a living in an honorable place in society at the same time. You will get paid much more in another profession, whatever your level of intelligence. So choose science only if you really, really love it. Don't go in for what's in fashion. Don't go for what the department seems to need. You're the one doing the work, so follow your heart, your interest in choosing the field of science. Peter mentioned this morning my coming to him to lead me. Let me say, first of all, a few things just to make clear the table. Baruch was Peter's first student, not I. Bob, though he wasn't the first author on some of the papers, because in those days they were in alphabetical order, but still Bob was my manche, how do you say, advisor in my postdoc work with him. And his name was on the first in the Nature paper and should have been in the Journal of Physiology, but they didn't do things that way then. Number 3, Duby was certainly doing perceptual learning before Meirav came to me and said, you know, I participated a little bit in perceptual learning experiments, and the one thing I noticed was that they show me this thing on the screen and I don't see anything. And then a little while later, suddenly I notice and I know what they're talking about. That's what I want to study. And that's what we began studying, which was perceptual learning. And luckily, we weren't really aware of what Duby was doing in parallel, so we used different approaches, got different results, and it's a good thing that we used a different string in the bow. Okay, let me go on. There are 2 paradigms for doing science. They sound very much like Treisman, 2 kinds of visual search. One is focusing attention to one problem, and the other is spreading attention to many. And the problem with focusing attention is that even you're using one methodology, studying one preparation, and let me just say that focusing attention to one problem methodology is very often chosen by physiologists because they have to use a lot of energy to learn to do all this physiology. I mean, they have to be surgeons, and they have to be animal trainers, and they have to be computer experts. But there's a danger of forgetting the big picture. The second method is spreading attention to many issues in the field, going from problem to problem, from methodology to methodology. This is often favored by cognitive psychologists who use similar psychophysical, sometimes fMRI techniques for different studies, and therefore they can go from problem to problem. The danger is insufficient depth in each issue. Even good results in disparate studies may not lead to being known for belonging to one result. My career has been more of the second paradigm, going from, as you've heard a lot today, from physics to biophysics, transduction to physiology, barnacle and cat to monkey and human, single neuron to behavior and psychophysics, automatic receptor field processing to perception and cognition, learning and conscious vision, categorization, multimodal interactions, but I should stop. And even studies with Marina on neglect syndrome and with Itai Chovers on macular degeneration. Nevertheless, let me say that I'm most associated with my work with Meirav over the last few years on perceptual learning, that's about 20 years, reverse hierarchy theory of perceptual learning, about 15 years, and reverse hierarchy theory of conscious perception, which is about 10 years old. Okay. And what I'd like to do now is to give a few examples of what made science exciting to me. Because I think my main message to a young scientist is do things which are exciting to you. And that's the only way it's going to work. So I want to go through a few experiences of mine. And this is, I don't want to tell you the science. I want to tell you the episodic memory, the feeling that there was in doing science and discovering things. The first, we heard some of it this morning from Baruch. This is, I'm sorry, I'm getting mics mixed up. Do you hear me now? Yes, good enough. Okay. So Peter, I think it's a rather old picture, and Baruch. We asked the following question. at one point, and Baruch gave some of the background, but I want to pinpoint to one question which we asked this morning. And as Baruch said, we sat together and we thought, and then we did the experiments. And we sat together and we asked the following question. Is phototransduction, is the whole process local, or is it spread out? For example, maybe the light-absorbing pigment itself is an openable channel. After all, that's what Huxley tells us about gating and so on. There's a channel, and whatever does it, in that case it's voltage, in our case it's light, should open that channel. Maybe it's local. And the other possibility is, no, there's secondary messenger and so on intervening. And we had 3 tools at our disposal. 2 transduction mechanisms, one turning the response on and one turning the response off. We had early receptor potential measurements reflecting the pigment state, so we knew exactly what was going on. And we knew there were 2 stable states for the barnacle, the rhodopsin and the meta. So we had this question, we had our tools. And then we said to ourselves the following. If it is a channel, like a door which opens and closes, there's one thing you can't do. You can't go to a closed door and close it. So therefore, if you transfer pigment from metarhodopsin to rhodopsin, which is supposed to close the door, if it's a channel, nothing's going to happen because it's already closed. On the other hand, and therefore when you later transfer it in the other direction and you open the door, it'll open. But if there's an intervening substance, which is spreading around and long-living, then when you move from metarhodopsin to rhodopsin, then you've created something which can close the door. But since the doors are closed, it's just going to hang around. Then when you open the door, it'll close it. So in the one case, you end up with open doors, and the other case, you end up with closed doors. Simple question. That afternoon, we did the experiment. And 2 hours later, we knew the answer. We had the answer, spreadable second messenger molecules intervene. I think that picture is, for me, the physics approach to biology. You ask your questions sufficiently tightly so that you can do one experiment and you know the answer. Yes, I know, Baruch, in biology you have to do a number because maybe this barnacle is different from that barnacle and so on. But, yeah. Okay. Our next one with Bob. And Bob said something about our discovering the mechanism underlying the Y cell nonlinearity. And we had recorded from lots of neurons and found that sometimes there was, even the nonlinear cells, sometimes there were 2 peaks when you turned light on and off, like that, on different parts of the receptor field. Sometimes there was 2 peaks, and sometimes there was 1. Okay? And the X cells always had only 1. Okay? But Y cells were confusing, and then we got different responses. One day, this is my recollection, Bob will see if you remember it this way too. We put out on the table, there was a big table there at Rockefeller, and on the table we put out all the responses that we recorded. We had this paper recorder and we looked at them and suddenly we saw the pattern. Responses at low spatial frequencies had 1 prominent peak, perhaps a small secondary, and at high spatial frequencies they had 2. Then we saw that the more the double peaked the response, the less it was phase dependent. So this is the picture that came out, and you can see there are, for the Y cell at low spatial frequencies, a lot of 1 peak, only at the null is there a double, and at high spatial frequencies, there are 2 peaks all the way across. That told us the answer, and many cells later we were able to develop the model. So I think you're getting that there's a moment of insight which tells us something which is exciting. Sima Schechter and I asked a different question. Ungeleider and Mishkin and Hubel and Livingston had said there are separate pathways, and Udi spoke about it today also, separate pathways for perceiving motion, color, shape, maybe other things. If that's so, then motion correspondence should be independent of other features. What do I mean by that? Well, as Shimon Ullman had shown, if you put up, the first scene is the green ones, the second scene is the red ones. So this will turn this way because this is closer to that. What about if 2 of them are triangles and 2 of them are circles? Will it turn to the closer or to the same shape? If shape has nothing to do, if it's a separate pathway from the motion pathway, then shape should not influence the result. Simple question. And what Seema and I found was that shape does have an influence, and you can actually titrate. You can do this experiment with bigger or lesser, put it at 45 degrees or 30 degrees and so on, and you can titrate between them. And therefore, you can give an equivalence measure of different shapes. You can give an equivalence measure of different contrasts, of positive versus negative contrast, of different sizes, etc. Okay, so we did a whole slew of experiments which were able to give us this answer. So here what I'm saying is, don't take the dogma for granted. Everybody said that they're separate pathways. Don't believe them. Test it and find out. Ask the right questions. Get a clear answer. Okay. Next. This was a period at MIT where I worked with John Mansell in the lab of Peter Schiller. I put up the picture also of Tommy Paggio because I spent half my time there at the AI lab learning a lot about theoretical things and theoretical approaches, and computational approaches, let's call it. And John Mansell and I asked the following question. V4 neurons have responses and are tuned very often both to color and to orientation. What if we got a monkey to pay attention, to do a task where he had to pay attention to color? Would the tuning curve to color become narrower and the tuning curve to orientation wider, let's say? And then when he paid attention to orientation tuning would get narrower and color wider. And so we put up the following stimuli, 16 stimuli, different orientations, different colors, and there are two kinds of tasks. This is an orientation task. He's told, find a grading which is vertical. Is it this? No. Is it this? No. Is it this? No. Oh, yes. Response. This tells him, look for the color red, irrespective of orientation. Is it this? Is it this? Is it this? Yes, it's this. Response. Okay. So we could switch back and forth. Well, indeed, some neurons tightened tuning for the intended feature, and some did the opposite. On average, there was no change. So we reported a negative result. And John actually did the same experiment again a few years later. He didn't trust me. I was a novice. So he did it with someone else, and again, with the same negative result. Why do I tell you this story? Because even though there's a nice idea, beautiful idea that tuning should turn this way and that way, if it doesn't work, it doesn't work. Give up and go on to the next thing. Take your next string in the bow. The other thing is, maybe we did it wrong. Maybe we weren't careful enough. Maybe the task wasn't difficult enough. There are all kinds of issues about how we did it. And maybe one should go back one day and look if it's true. Okay. Next, Meirav and I asked, what is the rule, the mechanism of transfer versus specificity of perceptual learning effects? And Duby has brought new ideas this afternoon. Let me stay where we were for a moment. And because actually what was going on in the literature was there were reports of this and of that. Sometimes there was specificity and sometimes transfer. For those of you who are not in the field, I want to say something about what transfer means. When you teach a class of law students that they should really behave well with each other and help each other on exams and so on, you might succeed, but when they go out there to practice law, they're not going to. It's transferring from one situation to another, which is the most important thing in education. You want to teach kids to behave, then you want them to behave for the rest of their lives also when they're not in school. If you want to teach them how to use math, you want them to use the math also when they go to the store and they want to figure out how much is a 10% discount on 100 checkout, and take out their computer and start doing. So it's to use the tools that you're giving them into new situations. So transfer is extremely important. It's probably the most important thing in education. So what we were asking was, well, how does that work in perceptual learning? So we explored parameter space, meaning we tried more difficult, easier, differences of position, all kinds of things. And we finally found that transfer for easier conditions, longer times or easier discriminations or more consistent stimuli, any one of them, as long as it was easier, then we had transfer. And if the conditions were more difficult because they were very short presentation times, or because the discriminations were more difficult, or because the stimuli were less consistent, then it was more specific learning. And we argued that transfer can only occur if the same neurons are involved in both tasks. You can't teach one kid something and then expect his friend who stayed home today to act better. It's only the kid who's there, okay? So you have to teach the neurons which are going to actually do the next task. So the neurons have to be broad, otherwise they can't do both. So we assign transfer to high-level, large receptive field neurons and specificity to lower cortical level neurons, which constrained receptive fields. We also found low-level learning depends on prior high-level learning and guidance, the reverse hierarchy of perceptual learning. I want to say something about a study with Nava, who presented wonderful things today. Does the brain really compute this very, very elegant intersection of constraints model for complex motion? I'm sure even at the ICNC, you still learn the intersection of constraints model for complex motion. And one of the proofs brought by the authors at their Pontifical Academy report was that if you have a triangle where all the lines of the triangle are slanted up to the right, all three, then motion of any line, which is going to be perpendicular to the line, each one of them has a downward component, even when the triangle is moving straight to the right without any downward motion. So if we perceive this triangle as moving to the right, that means we cannot use just the motion of the lines to compute it. And therefore, they presented another computation, the intersection of constraints. And Nava said, nonsense. The way we see this moving to the right is we look at the points, the corners of the triangle. There's no question about the way they move. When this thing moves to the right, those corners move to the right. That's not ambiguous. So all you have to do is cover them. Leave the three lines without the points, and then you see it moving down to the right. You take the average. You do not take the intersection of constraints. And Nava went on to show this in many different ways, and went on also to theoretically show that we cannot compute 3D motion just by using this kind of a computation. Okay. So we found that motion perception depends on unambiguous motion of points, corners, intersections for vertical motion when present, and on average motion otherwise. Exciting stuff. The next study I want to mention is one with Danny Amit, who I'm so sorry is not with us, because, and it's because of the ICNC that I did collaboration with him. And Volodya and Tanya came to the lab to work on monkeys. And the study I want to mention is actually Volodya did. But Danny pushed us to ask questions which were related to his favorite neural network theory and attractor networks. And so we understood, and other people had found, that in order to establish, well, let me just say first, delay match to sample task. I think I can give the example. Here it is. Delay match to sample task says you show a picture, show another picture. Ultimately, the monkey has to say, I've seen this one before on this trial. Okay? And then you do another trial with the same shapes, and he's got to remember them. And the way we understand that, not only we, but everyone around, is that when you turn on a stimulus, the response stays up, reminiscent of the PDA, Baruch, and there's a long delay activity. And when the stimulus comes on again, then there's some sort of comparison going on there with the delay activity that's present. And if there's no delay activity, then you know you've never seen it before. Okay. But we know that it takes about 100 viewings of that same picture to develop delay activity. Delay activity, according to the theory, says there are a bunch of neurons. They all respond to this. But when you give... I turned it off. Okay. When you give the same stimulus again and again, the synapses among these neurons get strengthened. And ultimately, they're strong enough so that when you give the stimulus, you not only turn them on, but they now activate each other again and again and again. You get reverberations and you get this delay activity. Okay. But it takes a few hundred, or 100 at least, stimuli in order to get this kind of response. So we ask the following question. What will happen if instead of using stimuli that the monkey has learned, we take novel stimuli? And we found 10,000 different pictures. Volodya found 10,000 different pictures on the internet and said, what will happen with these? And we know there's not going to be delay activity, so the monkey's not going to be able to do the task. Of course, we were wrong. See, no delay activity. How will he know that this was seen before? It's a novel stimulus, seen in this trial, never seen before. And what happened was that with novel images, they were much better. And that's because they used familiarity memory rather than working memory. And what that meant was that the first step towards getting this delay activity was strengthening the synapses just a little bit. And when you strengthen the synapses just a little bit, then the response is just a little bit higher than it was last time. And that's enough to signal that I've seen it before. Okay. So that required ultimately just a little tweak of the neural network model to have familiarity as a step to the attractor. Once again with Meirav. Looking over a large group of perceptual phenomena, there were many puzzles lurking. Can one physiological picture account for many of them? First of all, the difference between rapid feature search, pop-out, and slower serial search, which Anne has mentioned. What sets the mode? Rapid perception of scene gist and much slower perception of scene details. If the whole is built from the sum of the parts, how can we know the whole and not the parts? And my favorite demonstration is the following. What's the difference between these two pictures? Quick? The dog and the flower, right? Instead of noticing the number of steps, the position and shape of this window, the position of this window, the position of the... So there are lots of differences. But those are differences in V1, while the differences of the dog and the flower are high level. And those are the things which we consciously perceived first. And this change blindness and repetition blindness and scene extension and the site of binocular rivalry and segregation, integration, there's lots of stuff. And as you've heard and many times, thinking over all these things, we suddenly had this eureka moment. And I think that's the moment that we live for when we do our science, to suddenly see that all these pieces come together. And we had in our background the reverse hierarchy of perceptual learning. So it was in a way easy to say that early perception, just like early learning, is high level, and later perception as later learning is lower. So my advice, physics approach to biology, investigate the entire stimulus space, don't take dogma for granted, ask the right questions. Good theories explain many phenomena, and learn while and from making mistakes. Still, there are important things that have not been said. And if I have a moment, since it's been mentioned that I learned Talmud well, I asked my daughter, who really learns Talmud, to teach me a little bit. And she told me the story of Choni, the circle maker. I'm going to say it in English, but you guys can read it in the original Aramaic. Choni asked the following question. Psalms says, we were, at the return to Zion, we were like dreamers. And he said, well, that must refer to the return to Zion after the destruction of the first temple. And the time there was 70 years. Dreaming is associated with sleeping. How can you sleep for 70 years? That was his question. Strange question. Okay. One day, he went journeying on the road. He saw a man planting a carob tree. And he asked him, in how long will it bear fruit? And the man replied, in 70 years. So he asked him, are you certain you'll be alive in 70 years? And he answered, I found carob trees in the world. Just as my forefathers planted these for me, so I too plant these for my children. Here too is a message for ELSC. The people who are working hard at ELSC to build a unity in a community and to build a building, I don't know how long it's going to take for the building to get built. But a lot of this work, he says 3 years. Okay, if it's 3 years, great. I'm just saying that we do the work also for the next generation. We plant carob trees. And every one of us has students in their lab. I know students are cheap labor. But we also train them and teach them, and we teach courses because we're preparing the world for the future. And okay. He goes on. Choni sat down, had a meal. Sleep came over him. He slept. As he slept, a rocky formation enclosed upon him, which hid him from sight, and he continued to sleep for 70 years. Okay, Choni was the scientist. Okay, Choni wanted to try something out, and he wanted to see what is it like if you go to sleep for 70 years and you wake up. Why isn't the world like this? Okay, let's do an experiment. What would happen? And as a good scientist, he did the experiment on himself. He went to sleep for 70 years. When he awoke, he saw a man gathering fruit of the carob tree and asked him, "Are you the man who planted the tree?" He said, "No, I'm his grandson." So then it was clear to him that he had slept for 70 years. Who's the hero of this story? Of course, it's the planter of the carob tree. Okay, he's doing the right stuff. Choni HaMagal is called the circle maker because one day he made a circle on the ground when there was a drought, and he said to God, "Give rain. And I won't step out of this circle unless you give rain." So it started drizzling. He said, "No, that's not what I meant. I meant rain." So there came a hurricane. He said, "No, I didn't mean that. I meant rain." So it rained. So if he could do that, he figured, well, he can sleep for 70 years. But it didn't end so good. He went home, found his grandchildren, and he said, "I'm Choni." But they didn't believe him. He went to the study hall, overheard students saying, "If only Choni was here, he answered all our questions, he really knew this stuff." So he said, "I'm Choni." They didn't believe him, and they didn't respect him. He weakened, asked for mercy, and he passed away. And Rava says, that's what people say, give me colleagues, friends, society. It's chevruta, it's a funny word. It can mean any one of these, or give me death. Okay. So Choni's experiment failed. And it's not so good to jump to the next generation. I'm not sure what that means for us here. I'm just saying, I'm still in the same generation. I'm here with you guys. Okay. But it does say to us that collaboration is the spice of scientific life. And I've mentioned the work with Peter and Baruch, which was so wonderful. Peter's been a collaborator and friend for over 40 years, and Baruch, who shared that initial excitement of killing the red tail. I've been very lucky to work with Bob, with Anne, collaborate, not discuss things with Mickey, speakers who have been here, who have had grants with. Sorry, Howard, I didn't find a picture of you. And then collaborations within the ICNC and ELSC. I think they made fun of me last night, about working with Daphna. Well, ultimately we did. And I worked with all these people in the ICNC and have papers with each. And it's been wonderful. And most important and most wonderful has been the work with students. First, this group of students who are now teaching at different places in Israel and abroad. Nava now is NYU, moved to Barcelona. The others are at different places in Israel. And I should mention also Orna Rosenthal, whose picture I also didn't find, who's teaching in Oxford, was at Birmingham, moved to Oxford. And I'm sure I've forgotten some people, so excuse me if I have. But another one of them was the Center of Excellence with the Israel Science Foundation, which gave us 7 years of 5 investigators, Meirav, Dani Amit, myself, Udi, and Rafi Malach from Weizmann. And we used to meet, this is a picture from Eilat, of some of us, and we met often. And the wonderful thing of having discussions, not with your own group, but with this expanded group of people, all of whom are working on similar problems and so on. Again, I want to mention specifically the collaboration with Volodya and Tanya that I've mentioned, and with Marina doing work at Levenstein Hospital. And now what? What's next? I think my time is almost over, so let me just say a word about where I would like to go at this point. You know, I just got a grant from the Israel Science Foundation. It's a small grant compared to the greats here, but it's enough to keep working, and they don't mind if I do not exactly what I wrote in the grant proposal. So, so what would I like to do? And I was taken by the original thoughts of Turing about what a Turing machine is and how it works, and his description of a universal computing machine. And certainly our brains are universal computing machines. And if that's true, then one of the tapes is the program tape. Well, I'd like to find that in the brain and to understand where it is, and what does it mean. I mean, for example, we go from task to task, and studies have been done about task switching, but they really concentrated on the cost of switching and why you become slower when you have to switch, not about where is the task switching taking place and how does it take place? What does it mean that we go from task to task? What does it mean that we go from language to language? Okay, and we're able to read different things where the same symbol, this samach, which is a letter in Hebrew, can be an O in English and can be the number 0. And how do we go between them when we have the same symbol having different meanings in different contexts? So, and I don't think that attention is the whole thing. So I would like to study the program tape, its cerebral site and cortical mechanism, and what it means to switch between these things. And I invite whoever is interested to join maybe a journal club to start with, to review what's been done and what we have to ask and what can be done in the future. And finally, I want to thank my partner of over 40 years, without whose support I could never have been a scientist. Thank you, Annette. Thank you. Thank you. I thank you all for coming to this party, whatever it's for.